Okay, and if you have attended the last session, you would know we will be holding one live interaction session every week where we'll be discussing a few things related to that week's topic. So last week we discussed the different topics under classification of living organisms and some sample problems related to that. And this is the second week, so we will be just taking up week two mainly. But other than that, since your assignment for week three is due, I think, day after tomorrow. So first of all, we'll be discussing some sample problems that might be useful in understanding or figuring out how you should solve problems like that. And then we'll also go through the questions for week one and week two assignment. And in case anybody has any doubts regarding those, we can go through that. And finally, we'll revise those topics and then we can move on to the next week topics in the upcoming week for week three live interaction session. So beginning with the sample problems for week three. So the first question is about how the genomic organization in prokaryotes is actually present. So if you look at the options, you will see here a nucleosome, chromosome, chromatin, and nucleoid. So when we talk about genomic organization, it basically means the DNA content which is present in prokaryotic organisms, right? So nucleosomes, as you already know, are the form of compaction or organization of the genome within the cells, but they are not present in prokaryotes. So nucleosomes, they consist of histone proteins and the surrounding DNA, which wraps around it, and that allows compaction of the genome within more complicated or in higher organisms. So that is what we consider to be a nucleosome. The second type of genomic organization mentioned here is chromosomes. So chromosomes, on the other hand, are the most compact form, which is usually found during cell division, particularly when the M phase of the cell is, when the cell is undergoing through its M phase. So that is the second organization. And this is also a higher form of compaction, which is observed in case of higher organisms, or in other words, we can say eukaryotic organisms. The third option, if you see here, is chromatin. And chromatin is the relatively unorganized form of the genetic content where it has not been condensed, and it is sort of a cloud, it has a sort of cloud-like appearance within the cell, and therefore we call that as chromatin. And the fourth option, which in this case is the right option, is the genomic organization of prokaryotes, whereby they have a undefined or a sort of unorganized or cloud-like appearance within the cell, which is the nucleoid. So the terminology, whenever a word is usually ending with oid, it usually is referred to a structure which is not very well defined. So if you look into words like embryoid or organoids, they are not, an organ is a well-defined structure with a differentiated fate, whereas an organoid is a mass of cells which recapitulates, or it brings forth the characteristics of the organ but is not as defined as it. So similarly here, nucleoid is a nucleus-like structure, which is a characteristic of prokaryotes, as you might already be familiar. The next question is about which of the following organelles has a double membrane. So here the options are endoplasmic reticulum, Golgi complex, chloroplast, and peroxisomes. So if we look at all of the organelles inside a cell, we will see that most of the organelles, they are, in case of eukaryotes, they are membrane bound organelles. But if we specifically go through the question, it is asking about organelles that have a double membrane. That is unique to only a very few organelles within the cell, majorly the chloroplast and the mitochondria, right? So here, if you see, this is the structure, or rather a section of the chloroplast, where we can see very clearly that there is an outer membrane. There is also an inner membrane, and inside the chloroplast, we can see the stroma, which is like the base on which these individual grana are present. Grana are like stacks of thylakoids which are connected to each other, and we can see that present here. And obviously, as the question is demanding, we can answer that this is a double membrane structure or a double membrane organelle which is present inside the cell. Similarly, one other organelle which also has a double membrane is the mitochondria. And obviously, this is it has actually led to a lot of speculation and understanding regarding the fact that the chloroplast and mitochondria are potentially derived from prokaryotic structures, which in this case, sorry, which in this case has brought about the endosymbiotic theory, where it was believed that these organelles are actually the result of engulfment of prokaryotic structures or bacteria, particularly photosynthetic bacteria and aerobic bacteria, to primitive cells that has given rise to eukaryotes. So that is also a theory whereby it has been considered to be the reason why we gradually shifted to a different type of organization inside the cell. So that is the second question. The third question here is the bond cleaved by lysozyme. Lysozyme, as you know, is an enzyme. And what is it responsible for? It actually cleaves the peptidoglycan on the cell wall of bacterial cells. So this is the bond that is, so the question is asking for the bond that is cleaved by lysozymes in the bacterial cell wall. And particularly this bond is present between two different sugars, which are N-acetyl glucosamine and N-acetyl muramic acid. So if we look at the options, here we have beta-1,3 glycosidic bond, beta-1,6 glycosidic bond, beta-1,4 glycosidic bond, and alpha-1,4 glycosidic bond. So one glance at the question will obviously be a little confusing, but if you have gone through the lectures and the lecture notes well, you will remember that the bond between these two sugars is a beta bond. So we have this picture here for reference. So just a second. Yeah, so here we have the glucnac or NAG residues and the NAM residues, and they are particularly connected by the beta-1,4 glycosidic linkage. And this is the exact site where the enzyme lysozyme is able to cleave. So the correct answer for this question is the beta-1,4 glycosidic bond. Moving on to the next question. The K and O antigens, which act as surface-associated virulence factors, are present in A, the lipopolysaccharide layer or the LPS layer, B, the lipoteichoic acid layer, C, the inner periplasmic space, or D, the outer periplasmic space. So here you can already see the answer. The correct answer for this question is the LPS layer. So if you look at a cross section of any of the cell wall of any gram-negative bacteria, you'll be able to clearly see these some of these layers. So you can see that the inner membrane and the outer membrane are interrupted or interspersed by the peptidoglycan, which is what we were just talking about. The lysozyme is capable of cleaving the peptidoglycan, and it is able to do so for gram-positive bacteria, which do not have these extra layers of the LPS and the outer membrane, and therefore it is specific to restricted to those class of or that class of bacteria. So when we talk about surface antigens, very obviously, as you can understand that if an antigen, when we say antigen, it is majorly a protein, and antigens are usually entities or structures which are recognized by antibodies. That is actually the reason why antigens are named so. It is because antibodies are able to recognize them. Now, if you have any a little bit of background regarding immunology, you will know that any antibody, it is only able to access structures or any entity or any unit which is present on the surface. Therefore, antigens are most or more often than not, they are present on the surface. And therefore, since the outermost layer out of the given options is obviously the lipopolysaccharide layer, as you can also see in this structure, it is usually the reason why these entities are present on the surface. And you can also see an expanded structure for the same, where you can also see that the O antigen has been indicated, and therefore these are surface associated virulence factors. When we say virulence factors, it means from the part or from the perspective of the bacterial cell, whereby it is able to cause disease, or it is able to act as a pathogen because of having antigens like this on its surface. So that is the answer to this question. And the next question is quite direct if you look into it. So it is asking about which of the following species has a linear plasmid. So if you have looked into the structure, or if you've looked into the definition of a plasmid in general, it is considered to be, it is usually abbreviated as cccDNA, which means covalently closed circular DNA. So it means that when we think about DNA, we usually think of like a helical structure, which is linear or which is straight, right? But when we talk about plasmids, they are usually considered to be cccDNA because they are closed, and it is said to be covalently closed because it is not it's not a temporary sort of or a weak bonding which is closing that DNA. It is a covalent linkage. It's just a circular DNA which is linked by a covalent bond, right? So therefore, the usual idea when we talk about plasmids is that they are covalently closed and they are circular in their structure. Therefore, when we talk about a linear plasmid, which means a straight plasmid, or a plasmid which is sort of, you can consider it to be open, those are unique to certain specific types or certain specific bacterial species. So in this case, the correct answer for this is a direct question. You just have to know the answer. There's no way to actually bypass it. It is only a restricted group of bacteria that have this sort of property. So this is something you'll have to memorize and just remember, because it's a unique feature. So the answer, as you can look here, is Streptomyces lividens that has a linear plasmid. Please interrupt me if you have any doubts. I'll try to help you out, or I'll try to bring more material to the next live interaction session. And you can also obviously share any inputs you have in the discussion forum if you require. The next question is about the process of plasmid isolation. So the question here is asking about the reagent which is used for deproteinization. So if you go through the process of isolating DNA, you will see that there are several very distinct steps. And among those, for deproteinization, which is basically when your objective is to get DNA, when you largely look at the cell, if you lyse a cell, which means if you rupture a cell, all of the content, that all of the belongings within the cell that come outside majorly consist of nucleic acid and proteins, right? Now, if your objective is to specifically get plasmids, you will eliminate the other components from it one by one. So first, you need to remove proteins. When we say remove, it means first of all, you need to denature them. You need to inactivate them. And then you need to separate them from what is your molecule of interest. In this case, that is a plasmid, right? And apart from that, if you are looking at cells that also contain genomic DNA, which is chromosomal DNA, under those circumstances, you need to eliminate that as well. So in this case, we are only being asked about deproteinization. So the answer to this question is the PCI method, which is phenol, chloroform, and isoamyl alcohol. Now, if you've read the protocol, you'll know that majorly this PCI, or this mixture of 25:24:1, that is usually responsible for resulting in a phase separation. When we say phase, it means organic phase and aqueous phase, right? So the phenolic content, phenol, or the organic phase, it settles at the bottom, and what it basically contains is all of the a cell debris. And when we say cell debris, it means that when you are rupturing a cell using SDS and NaOH, which is obviously the other another option present here, that is used for rupturing the cell or disrupting the cell membrane. So whatever constitutes apart from these molecules, which includes the cell membrane, any other organelles, any other debris or any other unnecessary junk part of the DNA of the cell, along with this proteins, they get denatured and they settle down in the phenolic phase. The upper aqueous phase, when we say aqueous, it basically consists of chloroform. So that contains the DNA, and isoamyl alcohol forms the interface or the separation layer between the two. So during the process of plasmid isolation, you will be able to remove or not remove, you want that, so you can call it extract or separate the aqueous phase, and then you can use that to carry out further processing steps to purify and enrich for your plasmid DNA. So the deproteinization is carried out by PCI method, and it's not just for plasmid isolation, even for genomic DNA isolation, PCI is used, and it's a long standardized protocol for doing this process. So we'll now move on to the next question. So just a second. The biomolecule which is synthesized inside the nucleus is what? So if you just read the question, even without looking at the options, you'll know that it cannot be a protein, right? If we think about the processes, the basic central dogma within the cell, it obviously has majorly three things. We have the process of replication, we have transcription and translation. Apart from that, there are also other processes, but the central dogma mainly looks at these three. So the process of DNA replication, it obviously occurs within the nucleus, right? Where DNA, which is already present, gets doubled or it gets copied, and daughter copies or replicated DNA is produced within the nucleus when a cell is making the decision to divide, right? So that is the process which is occurring inside the nucleus. Another process, the next one along the dogma, which is the process of transcription, is for, just give me a second, please. Sorry for the interruption. It was an important call. So coming back, we were talking about the processes of the central dogma. So there was DNA replication inside the nucleus, the process of transcription, which is the process of transcribing or converting the content within the DNA into RNA, mRNA particularly, which is decoding the information in the genes. And the third process is translation, where you translate the information in the mRNA into protein, which will carry out majority of the functional aspect within the cell. I'm saying majority because RNA also has a lot of roles to play inside the cell, and it's not just the mRNA. Of course, there are other RNAs as well, which will bring us to the answer to this question, which is there are mRNA, there are snRNAs, tRNAs, rRNAs, and many more. So if you look at the options here, you see histones, DNA polymerase, RNA polymerase, and tRNA. The first three options are referring to protein molecules, right? Histones are proteins which are responsible for DNA compaction. DNA polymerase and RNA polymerase are enzymes which carry out the processes I just described, replication and transcription respectively. So these proteins can only be synthesized inside the, within the cytosol, which is where a translation process takes place. So if you're talking about synthesis inside the nucleus, it has to be either the DNA or it can be RNA. So in this case, it would be the tRNA. And its function is actually in executing the next process, which is in translation. So tRNA actually brings forth a charged amino acid, which is corresponding to the codon, which is present in the mRNA. So it reads the mRNA and brings the cognate. When we say cognate, which means it means the corresponding or the appropriate or the correct amino acid, and then it links it together to form the complete protein. So that was the question. And then we talk about the major proteins that cause condensation of the eukaryotic chromatin. So I've already discussed this, but obviously, if you look at the options, the polymerases are not involved in that. Ribosomes are involved in translation process, which is the synthesis of proteins, and histones are obviously the answer to that. So histone, there are several different types of histones. What they do is they form an octamer, which means 8 histones are present. They form a structure or a sort of base, you can say, around which DNA is wrapped, and that wrapping creates a compact structure. So the uncondensed or the unorganized chromatin that we previously talked about gets compacted, and it becomes more condensed, and it can be accommodated within the nucleus when histones and other important proteins, which are chromatin premodelers, etc. It goes into the area of epigenetic modification of DNA is carried out to condense the eukaryotic chromatin. The reason this is important is because the eukaryotic chromatin is much, much larger compared to the nucleoid, which we talked about for prokaryotes, where the genetic content of those cells is much, much lesser. So it does not require this, this huge condensation process, and it can be accommodated within a small cell. So that was the answer. Then the cellular organelle that cannot be obtained intact during fractionation is A, endoplasmic reticulum, B, lysosome, C, peroxisome, and D, nucleus. So when we talk about fractionation, obviously it is a process which, fractionation as the term indicates, it is a process where you are fractionating or you are getting different parts or components of the cell, right? When you need to study them individually. So for example, if you want to study mitochondria, then you have to separate the mitochondria from the rest of the cell. So that is how you basically take the cell homogenate or the lysate, and you subject it to different speeds for different durations, during which you centrifuge that particular portion or that particular content of the cell, and you get some sort of cellular fraction or organelle. Now, the question here is asking about which organelle cannot be obtained intact. So if you look at these options, you will see that most of these structures are quite well-organized entities, but the ER, which is simply a membrane-like, very spread out structure, it's not a condensed or compact structure. The ER is majorly, it's surrounding the nucleus, and it gets fragmented when you are carrying out the entire homogenization and fractionation protocol. So therefore, this organelle, as you can see in this image as well, cannot be obtained intact. So this is the homogenate, which you are subjecting to the processing steps. So instead, what happens here is that first you get the nuclei and the cellular debris in the first step of fractionation. These can be obtained. Then we get pellet, which is very rich in mitochondria and chloroplasts. Then when you subject to this very high speed for approximately, this very high speed for approximately an hour, you will get microsomes, which are little, like you can consider them to be like spherical structures, membrane bound spherical structures, which are which are not just the cell's plasma membrane, but they're also the ER. So that can be obtained upon this fractionation process. And finally, you will get the pellet rich in ribosomes after centrifugation at very high speed for a very long duration. So the answer to this question is the ER. Then we look into The phase of the Golgi complex that sorts lipids and proteins is called A, the cis phase, B, the maturation phase, C, the formation phase, or D, the proximal phase. So as the question and indicates, and as you have already studied, the Golgi complex is it is named so because it's not one single entity. Like you can identify one mitochondria, but the Golgi are individual um, Golgi that have assembled together, and they are responsible for the entire vesicular trafficking and protein sorting process, which is an extremely crucial um, responsibility of the cell. So this arrangement, if you look here quite carefully, it will show you here is the structure and the ultrastructure. The ultrastructure is better explained. So here you can see the cisternae, the individual cisternae, the entire this is the cis medial and transgolgi network which is present here. So since the question is asking about the sorting of lipids and proteins, it will obviously occur to so the after the protein has been synthesized and it has been transported from the ER to the from the cytosol or the ER to the Golgi, it will go through these individual Golgi cisternae, and finally it will be sorted to whatever its ultimate destination is via the maturation phase. So that is the phase which is actually responsible for carrying out this process. So that's what we see here. Next question is asking about the orientation of proteins in the plasma membrane, which is predetermined by their orientation on the ER or the ER membrane or the nuclear membrane or the mitochondrial membrane or the vacuolar membrane. So if you just look at this diagram which has been shown here, you will see that since we are talking about sorting or vesicular trafficking, so proteins which are actually going to get sorted or which are destined to be present on the plasma membrane, they have to go through this vesicular trafficking or sorting process. So this sorting process obviously occurs through the ER, then the Golgi, and then it is finally sent to the plasma membrane. So here we are looking, if you look at the options here, you will see there is the nuclear membrane, the mitochondrial and vacuolar membrane, but obviously the correct answer is the ER, since proteins after synthesis will undergo the processing steps, and the entire time the orientation in which the protein will finally get positioned on the surface or on the cell membrane or plasma membrane will be determined at the level of the endoplasmic reticulum. Then moving on to the next question, we are asking the membrane carbohydrates are exclusively confined to which side of the plasma membrane? So this is a relatively simple question. So when we are talking about carbohydrates, membrane carbohydrates in particular, they are present to the side which is at the cell exterior or at the outer surface of the cell. The reason being that they are serving as recognition entities, which if you remember a previous question, we talked about antigens for bacterial cells, particularly for gram-negative bacteria, where the O and K antigens were present on their surface. So apart from that, with reference to a more higher order cell, a eukaryotic cell, for example, their membrane carbohydrates serve a sort of similar purpose. So obviously their location has to be on the exoplasmic space. So if we look at the options here, the cytoplasmic for a double membrane plasma membrane or a double layer or a lipid bilayer, the cytoplasmic phase is towards the interior, and obviously carbohydrates cannot be present in such a reducing environment. The exoplasmic phase is towards the outside. The core of the bilayer is hydrophobic, so it will not accommodate anything except for the lipids which are present, the fatty acids which are present. So the phospholipid bilayer, it has the proteins, it has the fatty acid chains towards the core, forming the hydrophobic core. So that will not consist of the carbohydrates, and obviously both sides are also not a feasible option. So exoplasmic phase is the answer to this question as well. So we'll move on to the next question, where we are talking about the marker enzyme. for lysozyme. So lysozyme, as the name indicates, it's responsible for lysis, right? And when we are having zyme in the name, it indicates that it has some sort of um, enzymatic activity to perform. So I'm saying enzymatic because it's majorly responsible for digestion or for breakdown of cellular components. So in this case, we are looking for a marker enzyme, right? Which will, like, if you have, um, say you are given 4 different test tubes, and you have to identify which of these components has lysozyme, which is mitochondria, which has the cell membrane, which has some other cellular compartment, say the nucleus. So under those circumstances, you need a marker, or you need some sort of definitive indication to know which component is present in which test tube. So then the marker enzyme for lysozyme in this case is sorry, it's supposed to be lysosome. I wrote it incorrectly. Please forgive me. So that would be cathepsin under this circumstance. So lysosome is the compartment of the cell which is responsible for digestion, and therefore cathepsin, which is an enzyme which is responsible for digestion, would be present within the lysosome. Lysozyme, just to clarify any confusion, I just noticed it now while I was speaking. So the lysosome is the cellular compartment which is responsible for digestion of proteins and other components, and it has an acidic pH for that reason to carry out that digestion. And it's also responsible for breakdown of unnecessary or non-requisite components of the cell. On the other hand, lysozyme is the enzyme which digests the NAGNAM, the glycosidic bond between NAGNAM residues, as we've already discussed, that are present in the bacterial cell wall. So please don't confuse the two. This is an error on my part. Then we come to question 14, where we are talking about transferrin. So transferrin is a soluble carrier protein that transports iron through the blood by a process called as A, phagocytosis, B, pinocytosis, C, receptor-mediated endocytosis, or D, exocytosis. So the answer to this question is receptor-mediated endocytosis. So these are each of these processes are responsible for different functions. So phagocytosis is usually to take up food, or it's the process of engulfing something as that will be utilized by the cell as food. While pinocytosis, it literally, if you just want to uh, broadly understand the process of pinocytosis, it's the process by which a cell drinks water. So it's the process of taking up water from the surroundings for a cell. On the other hand, exocytosis means you are sending something out of the cell, right? But if a protein like transferrin in this case, it's a soluble carrier protein, what is it doing? It is transporting iron through blood. So it won't be sending out, or it won't be exporting, so to say, iron into the surroundings, but it will actually be responsible for bringing iron into the cell. So that is transferrin, which is acting like a receptor, and it is endocytosing, or it is taking up iron into the cell, and therefore the process would be receptor mediated endocytosis. So that's the answer. Then the next question, the lumen pH of the late endosome, where the contents are digested and delivered for further distribution, is A, 4.5 to 5.6, 6 to 6.2, 2 to 3, or 7 to 7.6. So this is also a very direct question, but if you have any confusion, you can just remember the fact that whenever you are trying to digest or break down any cellular content, you need acidic pH, right? Think, for example, your stomach, which is responsible for digesting all of the food that you consume. So that needs an acidic environment to execute that entire process of breakdown and digestion. In this case, we have also previously talked about how the lysosome, the suicide bags of the cell, or the digestive bags of the digestive bags of the cell, are also responsible for the same function. So the pH is always acidic under such circumstances, and therefore, but if you look at the pH 2 to 3, that is extremely acidic, which is not the kind of pH that would normally be accepted by a cell, because the physiological pH, if you remember, is 7 to 7.4. Therefore, a pH as low as the acids, like for example HCl or H2SO4, these extremely low pH is actually very hazardous to the cell. So even if it is membrane bound, a relatively acidic pH is of 4.5 to 5.6, but this is still a quite direct question that you have to tackle directly. Then we come to question number 16. So, when we are talking about the decision to divide, the question is asking, when is this decision to divide taken during the cell cycle, right? Or which phase of the cell cycle is this decision actually made? So if you remember the cell cycle, there is the G0, which is the quiescent phase. Then there is the G1, S, G2, and the M phase, right? The M phase is where the cell is actually dividing, right? Apart from that, the G0 is the quiescent phase. So if we just go by the process of eliminating incorrect options, we are looking at G0, the quiescent phase, so that's not where the cell is dividing. It just goes into a sort of dormant state under those circumstances. The G1, G2, and S phase are our remaining options. Now, S phase is where the cell is synthesizing or doubling all of its it's the synthesis phase, but it is doubling all of the cellular contents during that phase. So that is not when a cell can actually decide to divide. It is already in the process of preparing to divide in the very near future. So the decision has to be made before that, and therefore G1 is the phase where the cell is making the decision that, okay, now we are going to divide. The cell has grown enough and it is now going to undergo division and produce two daughter cells. So the decision for the same is taken during the G1 phase. And obviously the G2 phase comes after S phase, where it acts as a checkpoint to ensure that all of these cellular contents have actually doubled, and that there is a doubling of say the nuclear, the genetic material majorly, but also of other organelles, including the cell membrane, the lipid bilayer, so that the cell can now undergo division and produce two new daughter cells. So the G1 phase is where the cell decides that it is now going to divide. The next question is about histones, and when we are trying to figure out where or which phase are histones synthesized in, as we have already discussed that each of the phases are responsible for making some decisions. So the M phase, G0 phase, S phase, and G2 phase each have different functions, but since S phase is the synthesis phase where the cell is doubling all of its contents, it also has to double histones, because after the cell has undergone the process of dividing, which is during the M phase where it undergoes mitosis or meiosis, after that it again has to undergo the entire process of compacting all of the genetic material within the cell. And for that process of compaction of the chromatin, it requires histones. So histones also need to be produced and need to be doubled, and that occurs during the S phase. The cells which do not divide, once they are differentiated and they enter into the G0 phase, is option A, lens cells, option B, lymphocytes, option C, epithelial cells of the intestine, or D, the smooth muscle cells. So this question is basically asking for end differentiated cells, right? So if you just if you're not aware of the direct answer to this question, if you're trying to troubleshoot and trying to figure out an answer to it, you have to look for cells which cannot be replenished, right? Because they are, once they are gone, and you cannot have a restocking or replenishment. So the first option to be eliminated would be the epithelial cells of the intestine, because if that was the case, if somebody suffered from any sort of insult or any sort of disturbance to their epithelium, or to the intestinal epithelium, to their gut lining, basically, which occurs during a lot of diseases. Like, for example, if you have heard of people with epithelial with gut lining disorders, like for example ulcers, or some sort of cancer people are going through, majorly in the intestinal crypts, or colitis, then under those circumstances it would be very difficult for such people to survive, because the content which is retained within the intestine needs to be shielded from the remaining environment, which is into the entire body cavity, because those are very toxic materials and they should not be released. Option B. Hi Suchitra, but are you answering question 18? If you are answering that, it's unfortunately not the correct answer, and I'll address it why. So lymphocytes are the immune cells of the body. So if a lymphocyte has identified that there is some sort of a pathogen, say for example you have a viral infection, anybody has a viral infection and it has been recognized, then let's not say viral, let's talk about some bacterial infection. Somebody is suffering from cholera. Then once that particular bacteria has been recognized, the cell that has recognized this particular pathogen needs to divide. So they must the ability of the immune system to actually recover from any disease heavily depends upon the fact that there is clonal expansion, which means you learn more about it in immunology based courses, where the cell that has recognized the pathogen has to divide. So lymphocytes cannot be the answer, and so cannot smooth muscle cells. The answer to this question is actually lens cells. So lens cells, the cells in the eye lens, they do not divide, because imagine that, if have you ever heard of a person whose is expanding? If it did so, it would actually lead to a tumor, but that does not happen. So lens cells, they are indifferentiated and they enter into the quiescent phase where they do not divide any further. The next question is asking about which cells, during which phase of the cell cycle do chromosomes become highly condensed to prevent DNA damage and facilitate segregation to opposite poles. So as we've already studied the different phases of the cell cycle, and particularly if we talk about the M phase. So we talked about the cell cycle phases. In the M phase also, there are a lot of divisions. There is the prophase, metaphase, anaphase, telophase, and finally cytokinesis. So when we are talking about the process of segregation to opposite poles, that is obviously the very unique feature of anaphase only. So, sorry, yeah. So somebody joined, I think. Okay, so anaphase is identified by the V-shaped, or rather the process where from the metaphase plate, first the chromosomes are condensed during prophase. During metaphase, they are aligned along the metaphase plate, and after alignment, they are pulled apart by the microtubules towards the opposite poles, because the next step is actually to undergo cytokinesis, where one particular parent cell will be divided into two daughter cells. So anaphase has this particular unique criteria. So you actually need to keep in mind one unique feature of each of these cell cycle phases, so that you can recollect it when you are asked about it. So that's the unique feature of anaphase. And finally, for the last sample problem for week 3 is about the pH in mammalian cell culture media. So the question that might come up is, why is the pH so important? It is important because that buffers, you've answered B, yes, you're correct. So it is monitored by phenol red, and that is very important because it for mammalian cells to survive, pH is a very, very important criteria, and therefore you need to make, and it also it is also essential to make sure that there is adequate supply of nutrients to those cells, because they need to be replenished with fresh media containing all of those nutrients and growth factors periodically. So therefore, the indicator, which is the pH indicator, which is most commonly used for mammalian cell culture media like RPMI, DMEM, etc., it is phenol red. So you've answered correctly. Now, we have discussed the sample problems for week 3. Hopefully, it will help you answer questions for this assignment, this week's assignment. And moving on to the previously submitted assignments, we're going to go through all of the questions one by one. In case anybody joins or they have some doubts, you can bring it up and we'll try to address it. So for week 2, the first question was about the central core of the earth. So in week 2, the first lecture actually stressed on what are the different layers of the earth. That we talked about the baryosphere, the lithosphere, and the pyrosphere. So the central core of the earth, where there are all of these nutrients, where the molten magma and all of the components are present, is the baryosphere, the core of the earth. It is attributed to the baryosphere. The second question was about the which of the following is not an experiment to disapprove the theory of spontaneous generation. So the theory of spontaneous generation just talked about how organisms just evolve spontaneously, or life originates out of nothing else, right? So since that particular theory did not have experimental evidence to support it, there were experiments that disproved it, right? So if you go through the lectures, then there was Francesco Reddy's experiment, where he took 3 different jars and he showed how organisms need to enter when he placed a gauze that was not occurring, and organisms need to enter to give rise to new organisms, which he showed using maggots. Then in Lazzaro Spallanzani's experiment, he worked with an S tube or S-shaped tube with a flask, and he showed that when it was broken off and there was access of the outside air which contained microorganisms to the hay fusion which was present inside that particular flask, only then could organisms grow from it. And similarly, Louis Pasteur's experiment also showed the same. Out of these, Robert Hooke did not show any experiments with regard to this. In fact, Robert Hooke is credited with something different entirely. He is majorly credited with observing cells for the first time. So he looked into cork cells, and he observed little compartments which were present like in hundreds of in numbers of hundreds, and therefore he observed cells for the first time. There were no experiments from his end to approve or disapprove the theory of spontaneous generation. So that solves the second question. Then the third question was about connecting links between arthropoda and annelida. So in the lecture, where there has been there have been evidences that actually support the concept of evolving, of changes over time, and among those, the idea of connecting links had actually come up. So there was peripatus, euglena, lizards, and seeds. So among these, most of them are actually connecting links, but when we are specifically talking about arthropods and annelids, peripatus was this is also a direct question. If you just go through the slides and try to brush it up again, peripatus is the answer to that. So direct question again. Then the half-life of U-38 or uranium-38 is approximately what number of years? So this also was quite a direct question, and it's a fairly easy question to answer as well, because if you just remember, like if you are given these exact options, you can see that there's only one option that talks about a few billion years. So that would help you answer it directly. But anyway, it's a fairly direct question that you just have to recollect and just remember for the time being. So yeah, so basically the whole idea came about to identify or determine the age of the fossils. So for that, carbon dating was done, or relative aging was done, and under those topics only, there was a direct mention of the exact number of years it takes for uranium-38 to degrade to half of its initial amount. And if you go through the lectures, you'll see how Professor Vishal Trivedi has explained that if you have 10 grams of this, 10 milligrams of this and 10 milligrams of this, it indicates that it has disintegrated to 50% of its initial concentration. And therefore you can say that, oh, the soil or the rock present in this particular area is that many years old. So that is how it is used for dating. Then the next question is also quite direct. So potassium, or rather K40, it disintegrates to form radon, neon, argon, or xenon. So it's also a direct question. Potassium disintegrates to form argon, right? Then the Cenozoic era is obviously the present era. You don't even need to go through the other options, because if you remember the geological time scales, then majorly time has been divided into 4 different eras, and among them the most it's not even the most recent, it is the current era, the one in which we are living. So that is the Cenozoic era, and this was also a direct question. So I hope you got these options right. Then come the colloidal aggregates of large organic molecules are known as coacervates, aerosols, protocells, or emulsions. So obviously, the correct answer to this was coacervates. In case there was any confusion about coacervates versus protocells, if you go through the lectures a little carefully, you will see that protocells or eubions are also considered to be coacervates only. Like within the, we'll actually go through the entire content of week 2 after we finish all the question discussion, maybe in like half an hour. So there it was clearly mentioned that coacervates that contain DNA or genetic material, those are referred to as protocells or eubions, right? So if you're just talking about organic molecules, large organic molecules, which are forming colloidal aggregates, then those would be coacervates. So that was the answer to this question. Then comes the highest level of organization in Celenterata. So this is actually a question from both week 1 and week 2, which is one of the reasons why you should always try to keep looking at the lectures for the next week as well, because it will pretty much help you. So in Celenterata, so we had discussed how there is a progressive evolution also discussed in this lecture, but we had also discussed with the last lecture as well that classification has been done in such a way that you're going from more primitive features or more primitive structures towards more advanced structures. So initially there was a cellular level of organization in sponges or porphyra, and then you gradually move towards more complicated. When we say complicated, we mean more refined or more well-controlled forms of organization, and that was seen in this case for Celenterata. It would be for tissue level. So there is a unicellular organism does not have any sort of organization in that sense, but then you move towards cellular, you move to tissue level, organ level, and then finally organ system level of organization. So in case of Cnidaria or Celenterata, you see tissue level of organization, and that was the answer to this question. And it's an amalgamation of the content from both week 1 and week 2. So you need to keep that in mind, that whatever you're studying in earlier weeks should also be retained, and you can revise as per your convenience. So that was that. Then comes question number 9, where we are talking about the forelimb invertebrates. So the forelimb invertebrates is an example of A, analogous organs, B, homologous organs, C, convergent organs, or D, coexisting organs. So yeah, so if you remember these sort of morphological evidences for which were in support of evolution. So there we discussed each of these concepts. So majorly organs can be analogous or homologous. So homologous organs are those which have undergone divergent evolution. So they have a similar, you can say, structure or origin, but what has happened over time is that they have evolved to perform different functions. So forelimb invertebrates is an example of the same, because while forelimbs in vertebrates and in like the flippers, etc., these are all organs that have had similar origin. Their function today is, or in the, or for that particular organism of which they are a part, in those organisms, they perform very diverse functions compared to what their origins were. In contrast, if you talk about analogous organs, those are completely the opposite. Those are, when we talk about analogs, it means that they had different origins, but they have over time evolved to perform similar functions, and they have undergone convergent evolution. So convergent organs are not really a concept, and coexisting organs either. So in this case, the answer was homologous organs. And actually continuing with the theme of the previous question, which of the following is an example of analogous organs? Then forelimbs and vertebrates are obviously not an answer to this, because it's just what we discussed for the last question. Then fourth, horns and tendrils in plants, not that either. Instead, insect wings and bird wings. So when you refer to wings, they are, you would think that insect and bird wings are similar, but in fact, they are not. They have different origins, embryonic origins, developmental origins, but they have evolved to perform kind of similar functions for that particular animal. Then hind limbs and vertebrates are also not an answer to that. So that's that. For question number 11, we were asked which of the following organs is an extension of the integument. So this is a direct question. If you study the lectures provided, then you have feathers, bird wings, insect wings, and skin. So it actually is an extrapolation of the previous question as well. So there you saw how analogous organs, insect and bird wings, they have different origins. So insect wings in particular are an extension of the integument. So that also is correlated to your previous answer. Then coming to the previously discussed topic of how over time organisms have gone from more simpler, less organized structures to more complicated organized structures. We also discussed how the heart, or rather the chambers within the heart, have also evolved with time. So initial heart had just two chambers, one auricle and one ventricle, but obviously that led to difficulties, because it could lead to the intermixing of deoxygenated and oxygenated blood, which you otherwise call as venous and arterial blood, which is obviously not desirable. So that is a simple two-chambered heart. Then it moved on to three-chambered, three prime, and finally to a four-chambered heart, which we know currently that having two ventricles and two auricles is the most, you can say, most advantageous or most developed or complicated, and the most suited for the process of double circulation that we humans and other mammals are also experiencing. And that's how that prevents any intermixing, and it ensures that proper functioning of the heart can be carried out very smoothly. But for amphibia in particular, if you remember, then the amphibian heart has three chambers, and that was the answer to this question. And this also is a question which is actually a part of both week 1 and week 2. So please keep that in mind and just revise your lectures whenever you get a chance to do so. So that's the question. Then we come to coelacanth. So we previously discussed about connecting links and how the peripatus is the connecting link between these two Annelida and Arthropoda. But when we are talking about coelacanth, it was an example of the connecting link between bonefish and amphibia. So these are just some examples which you have to recall, because these are very unique organisms that have provided instrumental evidence to the concept and to the idea that you cannot very clearly segregate organisms into different classes, or very clearly demarcate different classes or different groups. The reason being that there are some organisms which share characters of more than one group, and that shows that they have gradually evolved over time, and therefore they possess some unique features for both of them, and therefore you need to keep this in mind. So coelacanth in this case was considered to be the connecting link between bony fish and amphibia. So that was the answer to this question. Then this very interesting term of how ontogeny or ontology. Here it's referred to as ontology. In some places you'll find ontogeny as well. So ontology recapitulates phylogeny, and this very interesting phrase, not just a term, was coined by Ernst Haeckel, if you remember, where he said that even the far developed, so the ontology or ontogeny and phylogeny are two terms that you need to understand first. So when we talk about phylogeny, it means evolutionary relationship that over a period of several hundred of millions or billions of years, how it has evolved, that is phylogeny. Whereas ontogeny is the developmental process that that an organism undergoes. So he actually showed that how over the period of gestation, how a baby or a fetus develops. That is sort of reminiscent, or it recapitulates how our ancestors or how we as humans have evolved from the more primitive versions. So it is actually recapitulating how our ancestors used to be to what we are today, and the developmental process kind of is reminiscent of what has happened over a period of millions of years, and is can also be considered to be a supportive sort of thought process for the idea of evolution. So that was given by Ernst Haeckel, and it was a very interesting thought process, which obviously is there in the lectures. So that we saw. Then coming to Archaeopteryx feathers, they are an example of which type of fossilized remains. So when we have talked about the type of evidences that actually support the process of chemical evolution, we have also talked about paleontological evidence. And when we talk about paleontology, it basically means that there is some sort of fossil or some sort of remain which is present and which has been recovered, and it has been used to study the changes that have occurred over time. So there, Archaeopteryx feathers were particularly considered to be impressions, which are a subclass of paleontological evidence. So they are one type of fossilized remain. So that is the answer to this question. So we'll next discuss the week 1 assignments. Just please give me a 2-minute break. I'll just be back after drinking some water. Okay, so now we are coming back to the assignment for week 1, where first of all, there's a very direct question about who is the father of anatomy. So if you remember, we had also discussed this, that Galen was considered to be the father of anatomy. So that was so we had talked about a few eminent scientists and their contribution. It's there in the lectures as well, and it has been presented here. So he actually worked with a lot of tissues, and therefore he was given this sort of honor, and he has been coined or has been declared the father of anatomy. This is a direct question. So that was the first question. The second was about euglenoids and which kingdom do they belong to. So Monera, Protesta, Animalia, and Plantae. So obviously they are protists. We had also, I think, discussed about Euglena and its outermost layer and the structure of Euglena in the previous lecture. You can refer to the slides or to the video lectures if you need it. So the answer to that is Protestant. And also with reference to lecture 1, sorry, the question 1, you should keep in mind the contributions and the specific the terminology that is given for each of these scientists, because they all have individual contributions. So Particularly, you should focus on those that are mentioned in this question. So you can, if you are unfamiliar with, if any of them has not been mentioned in the lecture per se, you can try Googling a few and you'll see that there are like quite a few eminent scientists that have made very significant contributions. So you can refer to the sample problems and this yours assignments, and that should help you during your final exams for this course as well. So that was the question. And sorry. So the third question was about the correct order of classification. So we had actually indirectly discussed this in the last week's live interaction session as well. So this is also a very direct question. So obviously the highest order is the five kingdom classification, or rather other kingdom based classifications, but most accepted of them, of course, is the five kingdom one, where it includes Monera, Protista, Fungi, Plantae, and Animalia. So the downstream or the subclassifications after kingdom are kingdom, phylum, or division, which is often used for plants, but majorly it's just referred to as phylum. So kingdom, phylum, the class, order, family, genus, and species. And after that, we had also studied about how the genus and the species are majorly, when you talk about scientific names, the most accepted or the most usually used format of nomenclature is the binomial classification, where you majorly just mention the genus and the species of the organism. So please make sure that you remember this very clearly. If you need, you can make a mnemonic, which is useful for you, and try to avoid, like mostly people, if you are just in a hurry and trying to answer this question too fast, you might just get confused between option A and B. It's just different in one order, which is of class, and order that has been flipped. So be very mindful when you're answering questions like this, because you just see kingdom, phylum, and directly you go to something else. But oh no, actually it's pure different in genus and species as well. But just in case you are given two options which are extremely similar, but they differ by just one option, by one order, then you need to just be a little careful about that. So that would be my suggestion for it. And like for here also, I also overlooked the difference between the two. So maybe you can go ahead and do that. So that was this question. Then again, cannot stress enough on the fact that you should just keep in mind like some the contributions of some of the most eminent scientists, at least the ones which have been mentioned in the lecture and in the assignments and sample problems we're discussing for now. So the scientist who was most famously known for naming plants was actually Carolus Linnaeus. So Linnaeus was also responsible for the whole classification process. And Whitaker also, you can see that Robert H. Whitaker was responsible for, not responsible, he was credited for giving forth the Five Kingdom classification, which is obviously the most widely accepted format. So these kind of some fine details you should at least try to keep in mind, because that would be helpful in answering a lot of questions. So that's that. Then we come to the next question, question 5, which was about Vibrio and the shape of these bacteria. This is a very easy and very direct question. So if you remember, bacterial shapes are majorly of these types only. So there are spherical bacteria, there are rod-shaped, spiral, and comma-shaped. So Vibrio are the comma-shaped bacteria, while spherical bacteria are coccus, rod-shaped are bacilli, spiral are like spirulina, etc. And comma-shaped is the answer to this. So that is that. Then the next question is actually something we almost discussed in the last week. We had a very similar sample problem that we addressed. So there we had talked about extremophiles, right? So organisms that prefer to live in and that actually enjoy very extreme conditions, whether that is of temperature, pH, one specific nutrient, or some other extreme condition, which is not the most ambient temperature for any living organism to survive in. that class or that group of, we should not say class so loosely, that group of organisms are usually extremophiles, and extremophilic bacteria also had other uses that we discussed. Like, for example, the polymerases are usually taken from such extremophilic bacteria because they are useful in carrying out PCR reactions that usually require very high temperatures. So similarly, one question here in this year's assignment was also about bacteria that can be found in hot springs. So obviously, hot springs are regions with very high temperatures. So, they have extremely high temperatures, which leads to the expulsion of water. So giving rise to the name hot springs. So extremophilic bacteria that would usually live under such conditions would be thermoacidophiles. Obviously, as the name indicates, they live in such extreme pH and temperature conditions. The other options are different. Here, psychrophiles are those bacteria that prefer to live under very cold conditions. So psychrophilic, so if you, so the basic idea behind keeping food inside a fridge and really like in freezers or in not just food, other reagents as well, but majorly let's just talk about food items. If you leave something outside, it gets spoiled because of putrefaction or spoilage by bacteria, majorly by bacteria. On the other hand, if you keep something in the freezer, it stays better for a much longer period of time because most bacteria are not able to survive under such low temperatures. They need an ambient temperature to actually be able to survive and to cause any sort of spoilage. So, psychrophiles, on the other hand, belong to this group of extremophiles where they live under very low temperatures, which is like completely opposite to what the question is asking for in this particular case, right? So just be mindful of that. If you have a question which is asking for something completely different, if it is asking for, say, for example, those that can be found inside snow-capped, some regions, snow-capped mountains, etc., then that would usually have psychrophiles. Similarly, halophiles and mesophiles are also extremophiles, but they are not temperature dependent. So your answer was obviously thermoastrophile in this case. Then in question 7, we are talking about organisms that are used in the filtration of oils and syrups. This was a very direct question, and it was also very unique features of chrysophytes. So this is something you need to remember as well. So there are chrysophytes, dianoflagellates, euglenoids, and slime molds. So this was incorrect typing, but anyway, it's a very, like, diatoms or chrysophytes, they are majorly used for this purpose, which is oil and syrup filtration. So this is where you need to be very mindful when you are studying, because under classification there are unique features for every class, every group. So that is something you just have to remember at least a few features because it be useful in this particular case. So that is for question 7. In question number 8, we are being asked about the correct scientific classification of Aedes aegypti mosquito. So here, if you look at the last two classifications, then you see that Aedes aegypti is common for all, and obviously Animalia at the beginning is also common for them. So now the remaining part comes is where you actually make the correct choices. So is it an annelid or is it an arthropod? So that's something you need to have a lot of clarity about because, sorry, excuse me. So Aedes mosquito or Aedes aegypti in particular, or rather in mosquito, it actually belongs to the group of arthropods. And if even if you're not sure about that, you would know that mosquitoes are insects. They belong to the Insecta class, so kingdom, phylum, class, order, family, genus, species. This is an actual class, Insecta, and therefore that comes under the phylum Arthropoda. So that is how you will narrow down your options. So once you have arthropod, insect, then you are left behind with just Animalia and just with option B and option D, right? Now, among these two, you need to remember that which one is like above, Dipterans or Culicidae. So Diptera is actually the order. So that is another very memory-based question, but it would get you quite far. And particularly, you can take this example and try to look for similar examples for the same. So that was question 8. Then coming to question number 9, which of the following are non-nucleated? So strange question. When we are saying non-nucleated, it's eliminating a lot of options. So we have bacteria, fungi, plant, and viruses. So obviously bacteria have nucleoid. We have discussed it extensively for week 3 sample questions as well. Fungi are also nucleated. Um, they can also be multinucleated, but plant cells obviously also have a well-defined nucleus. But the exception to this is the virus, which does not contain a nucleus. First of all, it's not really classified under living organisms entirely. They're considered to be on the verge of living and non-living, because they can be considered to be living once they have entered a host. But prior to that, they are not really considered as living entities. They are just considered to be um, particles, you could say. So they have certain features which they're not really connecting links, but they have certain features where they're truly able to replicate once they enter the host. So they are quite unique in their own sense, and they are non-nucleated, but they are a large number of viruses. They are considered to contain a nucleocapsid. So their genetic material is often part of a capsid structure, which is like a proteinaceous structure, which is the body, so-called body of the virus. So that is the capsid, and its genetic material is often wrapped around or wrapped within that capsid body. So it generates this sort of complicated structure, which you can consider to be a nucleocapsid, and that is majorly what the virus is composed of. Then we come to question number 10, which is about tapeworms. So if you remember, we had talked about how there were um, different groups. So we had like I gave you idea about platyhelminthes and ashtoninthes, etc. So plate-like structure is usually associated for flatworms or platyhelminthes. So tapeworms are also a part of, they are actually one of the classical examples for this particular phylum. So ashtoninthes come after that. They are roundworms. Then we have annelids, and then we have arthropods. So tapeworms, which are one of the major parasites for humans, they are actually a part of platyhelminthes. So it's a classical example, and you can just try and remember a few examples of the same. So that was question number 10. Then in question 11, we were asked which of the following is not an example of arthropods. So when you think about arthropods, it directly takes you to insects. So we have discussed mosquito, they're obviously arthropods. Spiders have jointed appendages, obviously arthropods. Cockroaches also have jointed appendages, head, thorax, abdomen is present. They're also arthropods. Leech, on the other hand, they are not. They usually think annelida. So their body, they have those sort of segmented body with like a parasitic mouth, a sucker via which they attach to their host, and then they suck blood, and obviously they're parasites, so they do not really belong to the class, to the phylum Arthropoda. So that was question 11. In question 12, it's a very direct question about an example of poikilotherms, so sea turtles for the answer to it. I won't stress on this much further because this is a direct question. We have Arctic fox, we have whales and penguins, but poikilotherms among them are sea turtles. So that was that. Then we come to question number 13 about tunicates and which subphylum they belong to. So if you remember chordata, there is a very detailed classification of chordates, which we had also discussed in the last week. So in the live interaction session for last week, and it's also there in the lectures as well. So tunicates belong to the subphylum of Urochordata. And then we also had other degrees of classification, cephalochordata, vertebrata, but obviously porifera is not a part of this. Porifera are a entirely different phylum, but yeah, this was a direct question. So you need to remember this. Um, and their body, the body of tunicines has been elaborated in question 14. So their body is covered by tunicine, which is made up of A, glucose-like material, B, protein-like material, C, collagen-like material, or D, cellulose-like material. So this is also a fairly direct question. And the answer to this is cellulose-like material. So that is how the body of tunicates are usually formed. And finally, we are looking at the final question for the first week's assignment. So salamander, it belongs to A, amphibia, B, contractiles, C, reptilia, or D, insecta. So the answer to this was amphibia, and this was also a direct question. So hopefully most of you got it right. And with this, we are coming to the end of the discussions for assignments. We'll just take like a 2-minute break, and then we'll just summarize what everything we had learned in in week 2. And if anybody has any doubts, you can try to bring it forth. And with that, we'll end the session for today, and then we'll move on to week 3 in the upcoming week. So just take a 2-minute break, and if you have any questions, please post them in the chat box, or you can ask, and then we'll move on to the summarizing of this week's topics, or week 2 topics, from the slides that have been uploaded in lecture notes on the board. So okay, so we will now try to summarize all of the topics for week 2. So I hope you've all gotten access. It's a open access presentation. So for week 2, there were 5 different lectures. Um, in the initial lecture, first, the structure of the earth. Okay, so first we talked about how living organisms can be studied majorly with respect to their shape, size, the spatial distribution, and their, of course, their biological activity, and the definition of life. But obviously the major question that was addressed here is that how did life originate, which was also the theme of this week, which is to study the origin and the evolution of life on Earth. So under that, they talked about the different layers, or the different you can say segments, rather layers is more appropriate, the layers of the earth. So under that, the discussion was on what is the baryosphere, the pyrosphere, and the lithosphere. So obviously, as you can see, the baryosphere, it's the central core of the earth, which has the molten magma, which is what gets expelled, sorry, when there's a volcano that is expelled from the center of the earth, with other metals as well, like iron and nickel, and it has an inner and an outer core region. Then the pyrosphere, the middle part, also commonly known as the mantle, and then comes the crust, which is the lithosphere, and lithos usually means the surface, and it's relatively less thick, and mainly it's very rich in silica and aluminium, which are some of the minerals that we mine for. Then came to the idea that for this earth, which was just made up of these materials and had this primitive sort of condition, what would be required to actually have life on earth? So there was some prerequisites which were taken into considerations to believe what was required for life to originate. So one of the first initial conditions which was believed to be required was a reducing environment where there was little to no oxygen. So that was actually that gave birth to the idea that organic materials are formed from inorganic materials. Then came the inorganic raw material for that life, the energy source like thunder, lightning, solar radiation, or other electrical discharge. And also, infinite time was believed to be required because there were no enzymes as such, because enzymes are proteins. So, if there is no life, where are proteins at all coming from? So, approximately a billion years would be required for the same. Then, several scientists, they came up with different theories for explaining how life has originated on Earth, which includes theory of special creations, which was believed to be majorly centered around religious concepts that God has created. God, in its own different format, has just created life as it is. But of course, there was no experimental evidence that backed it up. Then the theory of spontaneous generation, where non-living material directly gives rise to organisms. So while there wasn't much experimental evidence in support of it, as we've already discussed in today's session, there were experiments which disproved it, like Reddy's experiment, Spallanzani's experiment, and also Louis Pasteur's experiment. We'll come to those. Then the theory of catastrophism, which was also similar to the first theory and did not have much to back it up. Then came the cosmozoic theory, where they believed that life from other planets came to Earth. That is obviously also not possible to validate with any experimental evidence, and so that also did not was not considered to be substantial. Then the theory of eternity of life, similar, and finally the modern theory, which is the most accepted one and has a lot of um, experimental evidence and beliefs to actually support this particular hypothesis or theory. So it said that chemical molecules are actually giving rise to organisms as we know them. So coming to these, we talked about the first theory. Then came the theory of spontaneous generation. So that was actually quite debated, and there were evidences that actually went against it. So if you look at the claims, they by themselves seem quite outrageous about how, if you just dip a horse hair in water, it can't give rise to worms. So obviously there were experiments which went against it. So first was Reddy's experiment. We have talked about it. There were jars which were either kept open, covered, or covered with some sort of opaque material. And it was only seen that when air, along with microorganisms, had access to the interior of the jar, only then you can see meat decay and you can see organisms growing, right? So it couldn't just spontaneously evolve. Then Spallanzani's experiment also showed the same, how he validated the same findings as was given by Reddy. Not validated, he actually disproved the theory of spontaneous generation. And he just prepared a broth and he left it open versus when he left it sealed, he saw that organisms could not grow. Louis Pasteur did the experiment with the S-shaped sort of tube, and he showed how only when it could be broken off could you then see this. Only when it was broken off and there was air which could access the broth inside, only then can you actually find microorganisms growing. And therefore, he actually showed that it was not spontaneity, but it was something else. Some pre-existing life is actually giving rise to new life, and so he disproved this previous theory. Then came the theory of catastrophism, and obviously it was disproved as well. The cosmozoic, or the theory of panspermia, or the spore theory, was also not validated, where it was just believed that some sort of meteorite from some other planet, it just dispersed spores on the earth, and from there life stemmed. So there was no evidence, very strong evidence to back it up, and so this theory was also it didn't garner much attention. Then theory of eternity of life, it could not really explain much that where did it begin, right? If life has never started and if it will never end, it has to have a starting point. So there has to be some evidence to support that. So obviously it also got disproved as well. So, sorry. So finally, it brought us to the modern theory, the most acceptable form, which was discussed much more extensively in the lecture notes for the second lecture. So we'll actually go through that. So coming here, the modern theory, which was also known as the chemical or the theory of abiogenesis. It was actually put forward by two scientists who performed like some very major experiments. That was by Operin and Haldane. So there, they had made the a few assumptions. Just a second. Yeah, sorry. So there they had based their experiments on a few things that life cannot spontaneously come about. The environment of the earth would have been reducing, and inorganic molecules have actually given rise to the organic molecules, and solar energy or some sort of energy source is actually required for life to originate and propagate. So they gave experimental evidence for it, and they made a very extensive arrangement to actually support that. And this is one experiment you should study quite extensively. The setup, you will not necessarily need a diagrammatic practice, but it's a very relevant and very substantial experiment. So make sure you it's clear in your head. So there was the first most important, I would say, is this main setup, which has a glass chamber where there were these four gases, the mixture of these gases in this very specific ratio of 2 is to 2 is to 1 of methane, ammonia, and hydrogen, along with water that was, supplied as vapor from a separate setup. So this, along with electric spark, was allowed to mix in this glass chamber, um, which sort of recapitulated the condition on Earth back then. And from there on, when it was condensed, then like it was condensed to make sure that you get a liquid which you can sample, right? So it was passed through this over days, around I think 18 days. Yeah, around 18 days, it was allowed to circulate, and at periodic time periods, it was sampled, and then they looked for different compounds. And surprisingly, they could find a lot of compounds that were identified using chromatographic and calorimetric techniques, including a few amino acids, some nucleic acids, and also a few other sugars. So that actually gave idea that if you're getting some basic biomolecules, that will eventually be able to form more complicated not complicated, more more complex biomolecules, which would ultimately be essential for the organism to survive. So these conditions would actually, if they were validating the hypothesis that was put forward by Operin and Haldane. So they actually divided this entire concept into four, you could say, four major steps. The first one was that there has to be formation of inorganic compounds. So if you see, sorry, if you see here, then it said that um, the different energy sources, like we talked about volcanic eruptions or some sort of electric discharge from some sort of thunder, lightning, etc., or even volcanic eruptions, they provided energy which led to the formation of inorganic compounds. And then these inorganic compounds they were present in oceans, which is the dissolved form of those, and they gave rise to simple monomeric organic compounds. And that is what they had initially obtained with their experiment as well, like for example ribose sugars, or very small amino acids, fatty acids, glycerol, etc. And from there on, in the third step, they proposed that there would be more complicated organic compounds. So the simple monomers that they said would be obtained in step 2 would then aggregate or then cluster to form more complicated or more higher order structures. Like here you can see there's glucose, it's not a monomeric structure, right? It's made up of different entities, which is giving rise to one sugar, then ribose, glycerin, and so on and so forth. And in step 4, which is actually it's a bigger step. So they talked about the generation of molecular aggregates. Why did they give this idea? Was that since there were no enzymes present, if this is indeed the process that was followed, it would take an immense amount of time to form organisms as we know today. So molecular aggregates, in this case, they called as coacervates, they provided a more um, compact or condensed environment within which all of these reactions could take place, and they would be able to take in or discard anything which is required for their survival or not required for the same. So it actually accelerates the rate at which these reactions could occur, and it provided much better opportunity for the events to occur in the time scale that they have been proposed in. So these coacervates, they even further said that it divides and it produces smaller coacervates and so on and so forth. So here you can see that they are forming coacervates, they're dividing into smaller ones. And in fact, these are also like a subclass, or rather a more advanced version of these coacervates could be referred to as eubions or protocells. So these are coacervates that contain nucleic acids. So like we had talked about, the simple monomers, they formed like some sugars, ribose sugars, and you know that those give rise to nucleotides. So those nucleotides could eventually form nucleic acids upon aggregation, and therefore, when these first incidents of carrying genetic material could be seen, that was termed as eubions or protocells. Then, I'm sorry. Then then came the second step, which was the formation of the so-called first cells, where they found, apart from nucleic acid, they also found proteins and enzymes, obviously, which were very, very crucial towards the sudden acceleration of all of these cellular processes, because enzymes are obviously biological catalysts, right? They are capable of performing the conversion or the reaction involving the conversion of A to B in a much faster scale at an optimum temperature and a pH condition. They're able to execute that particular reaction. So these were actually considered to be the first cells on Earth when you have proteins and enzymes, and that is just speeding up the entire process of evolution many folds. So that is where they actually said that the first cellular form, so-called the first cell on Earth, is estimated to have appeared approximately 2000 million years ago. So quite a while back. But this actually summarizes the entire process and what are the proposed events that they believe that they hypothesized would have occurred to actually go from inorganic molecules and finally form the first cells to support their concept of bringing about the theory of the chemical theory of generating organisms. So this actually summarizes what was pretty much done. Then we'll come to the next lecture. So coming to evolution, the second part. So till now we have talked about the process of origin of life, the different theories which actually discussed it. Then in the second section of the content for week 2, there the concept of evolution was brought about. So the advancement of an organism over time, over a period of time, is the process of evolution. And one of the major contributors to this is Aristotle. So again, another eminent scientist that you need to keep in mind, because he gave the idea of scala naturae, which is the ladder of change, which is what you see on the right, like how organisms have gone from inanimate matter, and they have progressed through plants and other less complicated or more simple organisms towards more complex organisms, birds, then finally humans, and so-called perfection is achieved. So he said that this is how evolution has occurred. This was proposed by Aristotle. So evolution, the definition, as you can see here in red, is the process of changing from one form to another, and obviously that is occurring over a period of time. So unicellular organisms, then simple multicellular, and then it moves on towards more complicated multicellular organisms like plants, vertebrates, so on and so forth. So that was actually proposed, what was proposed by Aristotle. Then the question was that, is there evidence? Since we have been stressing a lot on actually backing up your hypothesis, which is one of the things that was previously also asked for when the origin of life was speculated by several scientists. So you need to support what you are proposing, or your proposal should come from some conclusions that you have derived based on your observations. So they actually had a lot of evidences in support of it. So first comes the morphological and structural evidence. We have discussed it. We have discussed a few questions actually related to it. So first is the body organization. So this we even saw in the content for week 1 as well, where we see that the complexity of organisms with respect to their body and how it is organized, that is also changing over different classes. So we have unicellular organisms, which are obviously, you can consider them to be the simplest. Then it goes towards the cellular organization, the tissue level, organ level, and finally organ system levels, which is something we have, like a circulatory system, a respiratory system, a digestive system, each having its own set of very clearly predetermined functions. So that is what we actually have discussed quite extensively. So here you see some examples as well, how amoeba has cellular level organization, whereas platyhelminthes and other higher organisms have this organ system, like I just mentioned. So you need to have that functional division of labor to make sure that an organism can function properly and smoothly. So that was there. Then comes homologous organs. We have discussed this as well. So they look different, perform different function, but they have similar origin, both topographic and embryonic origin with similar structure, but they are performing different functions. So they have diverged over time. So examples, this was also a direct question in one of the sample problems, the assignment problems. So we had the forelimbs in vertebrates, vertebrate animals like in man, cheetah, whale, and bat. They are performing different function, but if you look at their origin, it's the same. And also thorns and tendrils were also a part of that. So go through these lecture notes like quite clearly, so that these examples are very sorted in your head. The second type, or the second class of evidences, was for under morphological evidences, was for analogous organs. So they have the same function, they look similar, but their origins are quite different. So we had the example of insect wings and bird wings, where insect wings were integument extensions, which is also a question we have addressed. And it was a part of your assignment as well, whereas the bird wing is actually limb bones that are covered with flesh, skin, and feathers. So that is an example of an analogous organ that they are performing similar functions, but their origins are completely different. Also, you see here fins and flippers are another example of analogous organs as well. Then came the concept of gradual modification about the heart, and we have discussed that as well, how over a period of time it has evolved from two-chambered, where there was a lot of mixing of blood. Then came the three-chambered, a four-chambered, and finally the four-chambered heart. So in the question, we found amphibia. So there are actually three-chambered hearts, and there are pseudo four-chambered hearts in particularly reptiles, where it was also mentioned as three prime hearts. So in this question, we had amphibia, but you should have some recollection of each of these different types, because you can be asked from any of those. So anyway, that is actually supporting the idea that modifications are occurring over a period of time, and you can see them take place gradually. So that's what's there. Then came the idea of connecting links. So we had Euglena, Peripatus, we had these examples. So Euglena had plants and animals. Peripatus was arthropoda and annelida, which was one of the questions which we saw. If you want, try to go through images of these as well. That might be quite helpful in recollecting what are the two different classes or two different phylum within which it is considered to be a connecting link. Then came egg-laying mammals like platypus, we had discussed echidna. So they are also intermediate between the two. And also we should try and keep in mind what are some of the features which are the reason for which they're considered to be connecting links. Like, for example, if we talk about Euglena, it has characters of both plants and animals. So what are those characters? Try to just keep them in your mind. Then some other examples, Neopelina, Balanoglossus, we have discussed it extensively under hemichordates, how they are the connecting link between non-chordates and chordates. Coelacanth was also there in one of your questions, Phenodon, etc. So they are showing that organisms can evolve from ancestors that have characters of both of two different classes. Then we talked about how ontogeny repeats phylogeny or recapitulates phylogeny, the theory that was given by Ernst Haeckel. This also was one of the questions. And here he talked about the development of a frog and how it's the stages that it goes through, it recapitulates how it has evolved from its ancestors. Then we have also discussed one question on paleontological evidence, which is trying to study life based on the fossil records that we have. So what are fossils? How are they formed? What are the different types? What is the mechanism? Why is it at all occurring? And the different types of fossils. We had one question about Archaeopteryx feathers and what type of fossils those are. So you can find examples of those in this table. This is quite some extensive data, but just try to keep these examples on mind because they're really interesting and they provided a lot of evidence about how life has actually evolved over a period of several billion years. So in that particular question, we had Archaeopteryx feathers, which are impressions, but obviously there are other examples as well. So just, try to keep those in mind. Then the processes or the methods of determining fossil age. So obviously relative dating methods were previously used, but they are not, as the name indicates, they are not not as reliable as the more advanced techniques of absolute dating. So there is spontaneous decay, which can occur of radioactive nuclei, and that's actually used to study it. So we had a question on uranium, uranium and the lead technique of dating. So how it actually undergoes decay over a period of around 4.5 billion years. So that is one of the most, this and the carbon dating method, which was also there later, the absolute dating method. These are most commonly used. There's also uranium lead, which has been expanded here. It's the same slide. But yeah, then the carbon dating method, the potassium argon dating method was also there. This was also one of the questions. So as you can see, like assignment problems are just an example of what you can expect eventually in the final main course examination as well. So go through your lecture notes very well. These are quite detailed, but they're all very useful. Try to retain as much as possible. If you need, go through some mnemonics, which is like an easier way, which is helping you to remember maybe some very complicated idea or a lot of examples. Go through images and retain as much as you can, because these are useful techniques and they truly are important for understanding what biology is and what exactly is it we are doing. So that was the idea. Then coming to geological timescales, obviously using these fossils, the entire time period over the history of the earth has been divided into different eras. So we have Archaeozoic, Proterozoic, Paleozoic, Mesozoic, and Cenozoic era. So there was a question on this particular one. So modern era is obviously the Cenozoic era. And so another example of how you need to remember it. And this is just a slide which is summarizing what has already been discussed. And then we are coming to the mechanism of evolution. So in the just a second, sorry.
Ma'am, may I ask a question,
please? The mechanism of evolution. So we had the different theories that are put forward. So the first one is about how, which was given by Lamarck. So he talked about how evolution is occurring because there are needs. There is the need for new characters and those are inherited. So there was, he gave several on the part of Lamarck, he gave several arguments in support of it. He said how there is that organisms tend to become more complex over time. They use organs or they disuse organs, and that is resulting in evolution. And there is the environment in which that organism lives. And when it tries to actually adapt to that environment, then it acquires new characters. So that was the idea given by Lamarck, and he actually gave examples. One of the most famous examples is about how giraffes have evolved over time, and their neck has actually extended to accommodate the eating habits of the animal. So that was the example. He also gave several other examples about aquatic birds and how snakes, they have let go of or they have discarded organs they did not need, which in their case are legs, which are appendages. They did not need them, so they just over the period of time lost this ability. So that was his idea, but there are, of course, criticisms to this concept. One of the major criticisms, let me just expand this. One of the major criticisms was given by August Weismann, and his idea was about the continuity of germplasm. And he said that because organisms have somatic cells and germ cells, just because some changes occurring in one generation, it does not necessarily mean that it will be transmitted to the next generation. He actually proved that, where he used rats and he cut their tails over generations, and he saw that the next generation is not produced tailless. So it was a severely criticized concept, and there were other examples as well, like the boring of your ears, where you get your ears pierced in one generation, but the child never actually has pierced ears, and there were other examples as well. So it was severely criticized and disproved as well. So then Lamarck actually came up with a new concept, which was termed as New Lamarckism. And he said that, germ cells are not immune from the effect of the environment. They can carry the character to the next generation. But despite these concepts, he said that there are evidences which support that germ cells are actually responsible for carrying out these changes, and they are transmitting these characters over the next few generations, but he could not provide enough supportive evidence for this. So the theory of acquired characters, inheritance of acquired characters, was eventually not very strongly accepted. Then came the two very extensively discussed theories, which will be our last few minutes of summarizing. So the first theory was about natural selection. So it was proposed by Charles Darwin and Alfred Wallace. So majorly, it was also actually related to one of the questions I discussed in the previous session about the book which was written by Darwin, which was on the origin of species. And he actually proposed a theory of natural selection and gave the concept of survival of the fittest, that an organism which is able to acquire changes or acquire mutations that help it survive in the conditions that it is living in would be able to survive much better than another organism which is not able to accommodate or adjust to the changes in this new environment. So he actually studied that when he was on a world expedition on his ship in HMS Beagle, and he actually saw that. And he had actually several several inputs which he believed were supporting his theory, including rapid multiplication, that organisms are continuously trying to reproduce and they're trying to extend their race and they're trying to survive. So that was his first idea, and he gave examples of several animals. Second, he said that resources. So if every organism was capable of, limitlessly undergoing division, then there shouldn't be a problem. But the thing is that resources, or let's just put it directly, food in general is restricted. So the number of species effectively would remain constant, and therefore it actually leads to a struggle for existing. There it leads to competition. So there is food, water, and like there's a competition for food, water, and space. And as a result of that, there is within one species there is struggle. among different species also there is a struggle, and to adjust to the conditions of the environment, an organism needs to compete, and it needs to outcompete other organisms and survive better than that. So for that reason, natural selection or nature by itself would prefer organisms that are able to adjust much better to the environmental conditions, and would prefer their survival over those which are not able to adapt to the same. So that is supporting the idea of natural selection. Also, he talked about variation and natural selection, as I've just discussed. Uh, so inheritance of useful variation. So whatever variations you acquire, they can either benefit or they can be a disadvantage, or you can say they might not have any advantage. So the theory of natural selection actually proposes that whatever is beneficial will be retained, and the other characters would eventually be eradicated from the population. And finally, the formation of a new species. So because of the struggle for food and existence and the concept of natural selection, only individuals that are able to adapt will survive, and therefore it supports the hypothesis. And that's pretty much been validated here as well. And there were several evidences that supported this concept also, like, for example, for sheep that are shorter, since humans, for their own benefit, they choose to keep the sheep that are not able to cross fence and not able to escape. Since those are preferred over a period of time, such sheep have only survived. Similarly, mimicry and protective coloration. So if an organism is adapting a color which is allowing it to survive better in one particular environment, you can actually read about this in the concept of industrialization, and how the mammoths, how certain organisms, they change their color to adapt to the change in the environment. So that is very commonly seen. And this all sounds very perfect, but there were obviously criticisms against this theory as well. So there was the theory that why are vestigial organs then being present? If it is not in use, it should be eliminated. It should not be propagated over generations. Then no explanation for the variation. Then continuous, discontinuous variation, the pangenesis theory by Darwin. These were all um, debated quite severely, but the concept of natural selection provided a lot of background, a lot of, at least, starting ground for scientists to understand what is exactly happening over evolution. However, in complete contrast came the mutation theory by Hugo de Vries, where he actually said completely opposite thing. And he said that this process of evolution is not gradual, it's jerky, it's immediate, and it occurs suddenly out of like nowhere. There's suddenly a mutation and you suddenly see changes, and therefore organisms are different from their ancestors. So he performed some experiments where he took this particular plant, the evening primrose, Inothera lamarckiana, and he saw that majority, when he carried out self-pollination of these plants, of these wild-type normal plants, he found that a large part of them were normal, but there were some plants which were different. And when these different plants were self-pollinated, among them you could find this variation, but you could also find other plants which had much more variation. So he could find several different subclassifications from them. So he concluded that mutations are the major reason why evolution is occurring, that they occur out of nowhere. One mutation can give rise to a species, and several other conclusions based on that. So his idea was, it seems logical, because now we have more or less accepted that mutations are the reason these changes are occurring. And there were several evidences for this as well, about, for example, the Ancon sheep, the Hereford cattle, how the hairless cats, dogs, etc. can also be seen. And these were majorly accepted, but there was some evidence also against it, that if mutations were occurring that frequently and that regularly, then wouldn't you just be able to see a new species every day, because mutations are occurring that frequently, right? So it was debated. And another one major very debated idea was that if you're talking about mutations, if there is a normal allele or a normal gene which is present, then it would overpower the mutation and would allow the previous species to survive, because mutations are recessive in nature. and dominant mutations would bring about evolution. So Hugo de Vries did not provide much support or much evidence against or in support of this. So based on that, this theory was also, it remains slightly debated, but what you can effectively conclude from all of these different lectures is that there are, when you're trying to understand a scientific process, there is no predetermined idea. What scientists and what researchers in general do is they go through observations and they try to arrive at a conclusion, and there will always be arguments in favor and against it. So you have to defend that, or you have to accept whatever criticisms have come in, and then you build your theory further. So that's pretty much what the idea of science is, and that is how evolution and origin of life is studied, which covers our more or less the content for week 2, along with the assignment problems that we have discussed and some sample questions for week 3 also, which we have discussed. I will be uploading the slides and you will get access to it. And also the lecture would, not the lecture, the live interaction session would also be uploaded soon. So I'm assuming there would be no more questions till now. In case you do, please post them in the discussion forum. We'll get to it there. Or you can join for the next live interaction session next week, where we will be looking at more sample problems and assignment problems. And also we'll discuss the week 3 content, and we'll look into whatever anyone else has to contribute. So thank you for joining, and I hope you had a good time and it was useful to you. So thank you so much.